Good health is a state of complete physical, mental and social well-being. This means that it's not just about having an absence of disease. It also includes eating and sleeping well, getting regular activity and limiting the intake of harmful substances such as alcohol and drugs. Having good social well-being includes how well you get on with other people, and then good mental well-being includes how you feel about yourself. So someone healthy needs to have a combination of all three types of well-being. Here are examples of diseases, and they can be separated into two groups. We've got communicable diseases caused by pathogens. These are microorganisms that cause disease. Communicable diseases can be spread from infected person to other people. The other group are non-communicable diseases, and they're caused by either problems in the body such as faulty genes or lifestyle factors such as drinking alcohol, lack of exercise, poor diet or smoking. Non-communicable diseases can't spread from people to people. HIV is caused by a virus which destroys white blood cells in the immune system. People infected with HIV often develop AIDS, which is known as acquired immune deficiency syndrome. This means that they have a deficiency in their immune system because the white blood cells are destroyed. Because the immune system can't protect them from secondary infections, many people die from these secondary infections. So they're not dying from HIV, they're dying from the secondary infection because the body can't protect itself using its immune system because the white blood cells have been destroyed by the virus, HIV. An example of this is here where, as the number of TB patients has increased, the number of TB patients with HIV has also increased. So here's an example of a correlation between two diseases. And the reason why they're correlated is because having one disease sometimes increases the susceptibility of having another disease. Another example of this is when a disease damages the body's natural barriers such as the skin, and this allows other pathogens to get into the body more easily. Or a disease stops an organ system from working such as lung cancer, it makes other diseases more likely to occur such as pneumonia. So in all of those examples there is a correlation between diseases where having one disease means a person is more likely to have another disease. Non-communicable diseases can be influenced by lifestyle factors such as nutrition. This is a result of poor diet where a person is getting either too little or too much nutrients from their food. This is known as malnutrition. So even eating too much nutrients in food is also known as malnutrition, which can lead to obesity. Sometimes a diet could lack certain nutrients, and that means that that person is deficient. So they've got a deficiency in certain nutrients such as vitamins and minerals or even protein, and this could lead to non-communicable diseases. Another lifestyle factor that could influence non-communicable diseases is the effect of alcohol. Alcohol contains a chemical called ethanol, which is broken down by enzymes in the liver. The liver enzyme breaks it down into toxic products which are poisonous. Therefore, the more alcohol is consumed, there's a greater risk of developing diseases such as liver cirrhosis, and this is where the liver tissue scars. In your exam, you have to relate this effect on a local, national and global level. So an individual who drinks a lot of alcohol has an increased chance of having liver cirrhosis. This means that their family might have to support that person, and then on a national and global level there is a cost to society for treating patients with liver diseases as well. Cardiovascular disease is a result of circulatory system functioning poorly, and a sign of this is high blood pressure. Blood pressure is the pressure exerted by the blood against the artery wall. A high blood pressure is when there's more pressure exerted by blood against the artery wall. And this is most likely due to the heart contracting more strongly, such that there's more blood colliding with the artery walls, exerting a higher pressure. Eating too much fat increases the risk of cardiovascular disease. So if we want to find out who's at risk of getting cardiovascular disease, we need to be able to estimate how much fat that they have. And one way of doing this is by calculating BMI. This involves finding the mass and dividing it by the height squared. So in this example, we've got someone who's got a mass of 100 kilograms and a height of 1.7 meters. So their BMI would be 100 divided by 1.7 squared, which equals 34.6. This means that this person is obese and has a very high risk of cardiovascular disease. So a doctor might advise this patient to decrease their mass. If they decrease their mass to 70 kilograms, their BMI will now be 70 divided by 1.7 squared, which is 24.2. And this is a healthy weight. So using this example, you can see that decreasing the mass decreases their BMI, and this decreases the risk of cardiovascular disease. The problem with BMI is some people have got a high mass not because they've got too much fat in their body, but because they've got a lot of muscular mass. Abdominal fat is very strongly linked to cardiovascular disease. So instead of calculating BMI, another way of measuring the fat is by calculating waist to hip ratio. In waist to hip ratio, we can see a trend where as the waist to hip ratio increases, the risk of cardiovascular disease also increases. And this is because there are more abdominal fat in the person. So a doctor might advise a patient that they need to decrease their mass because that will decrease their waist to hip ratio, which will decrease their risk of getting cardiovascular diseases. To calculate waist to hip ratio is just the circumference of the waist divided by the circumference of the hip, where both units is centimeters. So example, a woman has a waist measurement of 29 centimeters and a hip measurement of 36 centimeters. So to calculate the waist to hip ratio is 29 divided by 36, which equals 0.81. So in this example, they've got a moderate risk of cardiovascular disease. As you can see from the chart, when you increase your waist to hip ratio, there's an increased chance of cardiovascular disease. Tobacco cigarettes contain harmful substances such as carbon monoxide, nicotine, and tar. The effects of these harmful substances include carbon monoxide reducing how much oxygen the blood can carry. So some of the red blood cells carry carbon monoxide around the body instead of oxygen, and this means that less oxygen reaches the body cells. Nicotine is a chemical that is very addictive, so it's very hard for people to stop smoking. And then chemicals in tar are carcinogenic. That means that they can cause lung cancer. Smoking is also a lifestyle factor that increases cardiovascular disease. So as we stated earlier, tobacco smoke contains harmful substances, and these harmful substances can damage the blood vessels. This causes fat to build up on the damaged blood vessel area, and when this happens, the blood vessels become more narrow. By becoming more narrow, the blood pressure increases, and that means that there's an increase in the risk of blood clots forming. And if a blood clot forms, that means it could lead to a heart attack or stroke.
high blood pressure, which can damage areas in your artery walls, making it easier for fatty material to build up over time. And smoking, where chemicals in tobacco smoke can also damage arteries, leading to this fatty material buildup. If a fatty deposit breaks down or ruptures, a blood clot can form around it. This can block your artery. If this artery supplies blood to your heart muscle, it can cause a heart attack. If this artery supplies blood to your brain, it can cause a stroke. There isn't a cure for atherosclerosis, but thanks.
So just as the video said, a blood clot that blocks blood flow into the brain could lead to stroke, and a blood clot that blocks blood flow into the heart muscle could lead to a heart attack. And this because these organs, or cells or tissues in these organs, won't receive nutrients such as glucose, and also they won't receive oxygen from the blood. And because the blood can't supply oxygen and glucose for respiration, these cells will die and they cannot function, and that could lead to either stroke if the blood flow is restricted to the brain, or a heart attack if the blood flow is restricted to the heart muscles. So to summarize this, substances in cigarettes can cause blood vessels to narrow, which increases the blood pressure, and this could lead to cardiovascular diseases such as heart attack or stroke. Now when you look at non-communicable human diseases, it's an interaction of lots of different factors. Not only cardiovascular disease, but also cancer, whether it's lung and liver diseases, or also diseases influenced by nutrition. So you can see on a global scale that in certain areas or certain countries, there's an increase or higher risk of non-communicable human diseases. And this is because of an interaction of these different factors, which are lifestyle factors. That could include how much exercise or diet you get, which has an influence on obesity or malnutrition. It could also be due to alcohol, which influences things like liver diseases such as liver cirrhosis, or it could be due to smoking as well, which has an effect on cardiovascular disease. So there's a combination of all these different factors. So we need to evaluate some different treatments for cardiovascular disease, and that includes 3 different types of treatments. So someone who's at a very high risk of getting a cardiovascular disease could have a treatment that involves changing their lifestyle. This means the doctor might advise them to have a healthier diet, to get more exercise, lose weight, or stop smoking, drinking. So exam question might be, explain why a doctor may advise a patient with a high BMI to give up smoking and exercise more. And that's because when they give up smoking, it reduces the risk of heart disease. Stopping smoking will stop harmful substances from tobacco smoke getting into the blood, and that which increases the risk of damaging to the heart and blood vessels. Whereas exercising more reduces the body mass, in other words, it reduces the BMI, and that reduces the risk of getting cardiovascular disease. The disadvantage of lifestyle factors is it takes time to work. So some people who have a very high risk of cardiovascular disease and need treatment straight away might take medication as well. And these medications are lifelong. That means that they're prescribed medicine that reduces blood pressure. But the disadvantage of these medicines is that they could have side effects. Those people with cardiovascular disease that are at very high risk of dying might then take surgical procedures, such as a stent or a heart bypass. For a stent, there is a narrowed blood vessel, and this blood vessel can be widened by inserting a stent, and that stent holds open the narrowest part of the blood vessel. And that means that there's an increased blood flow because the stent widens the narrowest part of the blood vessel. This lowers the blood pressure and decreases the risk of cardiovascular disease, such as heart attack or stroke. In a heart bypass, a person's got a blocked artery. So that means you can insert blood vessels from another part of the body such that the blood flow can be bypassed around the blood clot, and that means that those tissues after the blood clot can be supplied with oxygen and nutrients again so that they can still function. So this is an example of an operation that restores blood flow to cells or tissues beyond the blockage, because without oxygen and nutrients those heart cells will die and the person is very likely to suffer a heart attack. So to do that they need to have a heart bypass. So exam question, state and explain two methods of treating cardiovascular disease. If you have a heart bypass operation, it inserts new arteries into the heart that carries blood to damaged areas of the heart so the cells can still contract properly, whereas narrow blood vessels can be widened using a stent, and that allows blood to flow through more easily. The big disadvantage of surgical procedures is there's an increased risk of infection. In the exam you might have to mention the advantages of each of these cardiovascular disease treatment, which is the opposite of the disadvantage for others. So for instance, the advantage of lifestyle change is that there are no side effects or risk of infection, whereas the advantage of lifelong medication is that it has an immediate impact and there's no risk of infection. And then the advantage of surgical procedure again is it has immediate impact and there's no side effects. Pathogens are disease-causing organisms, including viruses, bacteria, fungi, and protists. There are different methods of spreading, so they can be transmitted through water, airborne, animal vectors, oral transmission, which means eating or drinking contaminated food / water, and pathogens can also be spread through the transmission of body fluids. So we need to know how this spread can be reduced or prevented. That could involve boiling water to kill the bacteria, or for airborne you could isolate the infected organism and that prevents the spread of pathogens. For animal vectors you want to kill the vector, or you could use artificial physical barriers such as mosquito nets. For oral transmission you can prevent the spread by cooking food thoroughly, and that will kill any bacteria that's in the food. And then for body fluids you can use artificial physical barriers such as a condom or even protected clothing to stop certain body fluids from being transmitted. You could also isolate an infected organism if there is a high risk of body fluids transmitting the pathogen. So for your exam you need to know a lot of information for all of these diseases. So cholera is due to a bacteria, the symptoms are diarrhea, is spread through contaminated water. So to reduce or prevent the transmission you could boil the water and that will kill the bacteria. Tuberculosis is also caused by bacteria and its effect is lung damage. It's spread through the air, so it's an airborne disease. And to reduce or prevent the transmission, you could isolate the affected individual to reduce the spread of the pathogen. Malaria is not caused by mosquitoes, and it's definitely not caused by viruses. It is caused by a pathogen called protists. Mosquitoes transmit the malaria disease when they bite a person who's already infected, and that means that they get the protists, and then when they go and bite a healthy individual they transmit the protists to that person such that the healthy person now gets sick because they've got malaria. So it's the protist that causes the disease, not malaria. Malaria helps transmit the disease from an infected person to a healthy individual. So in this example we've got a pathogen, a vector and a host. The mosquito is an animal vector because it helps to transmit the disease from one person to another, but it doesn't cause the disease. The disease is caused by the protist that is now in the blood. And then humans are the hosts because they're the organism that can be infected by the pathogen. So the pathogen that causes malaria is a protist known as Plasmodium. The symptoms is that it damages red blood cells and liver cells as well. It is spread through animal vectors such as mosquito, and then to reduce or prevent transmission you would want to kill the animal vector or use artificial physical barriers such as mosquito nets. The pathogen that causes stomach ulcers is a bacteria that causes stomach pain. It is spread by oral transmission, so swallowing food that's contaminated or drinking water that's contaminated. So to reduce or prevent the transmission, you would make sure that the food is cooked thoroughly or that the water is boiled. Ebola is caused by an Ebola virus, and the symptom is hemorrhagic fever. It is spread through bodily fluids. Because there is a high transmission effect, you would want to isolate the affected individual. Anyone who's trying to treat a patient who's got Ebola might want to wear a artificial barrier, which involves using protective clothing. And then cholera ash dieback is caused by fungus, and the symptoms include bark lesions. It's spread through the air, so it's an airborne transmission, and to reduce or prevent that transmission you want to isolate any tree that's infected by cholera ash dieback so that it doesn't spread the pathogen. And then here are some additional ways of reducing the spread of these pathogens. So in the exam you also need to know some common infections that wasn't mentioned in the previous slides. That also includes HIV, which we mentioned earlier, destroys white blood cells, leading to the onset of AIDS. And here we've got animal cells, plant cell, and a bacterial cell. This is an example of a virus, so it's not an animal, plant, or bacterial cell. And all viruses have two things in common. They contain genetic material, which could be RNA or DNA, and they also contain a protein coat known as a protein capsid around the genetic material. Animal and plant cells replicate by mitosis. And then you can see here, these are bacterial cells, and this bacterial cell also reproduces using a method very similar to mitosis known as binary fission. So how do viruses replicate? When viruses replicate, they need to replicate the genetic material, and they also need to synthesize proteins, because those are the two components of a virus. Problem with this is they don't have enzymes to replicate their genetic material. They also don't have ribosomes to synthesize proteins. So we can conclude that viruses are unable to replicate on their own. So if they don't have the processes to replicate their genetic material or to synthesize proteins, instead they inject genetic material into a host cell. And the reason why they do that is because they want to take over the host cell's processes so that the host can make new viral genetic material and new viral proteins. So because they lack the cell processes to make to replicate genetic material and to synthesize viral proteins, instead they get the host cell to do it for them, because the host cell has those processes. This means invading plant cells, bacterial cells, or even human cells, like in the case of HIV or Ebola virus. And that's because those host cells have the processes to replicate genetic material, and they also have ribosomes that can synthesize proteins. So this is how viruses reproduce. First, they take over a host cell by attaching to a specific host cell. Then they inject their genetic material into the host cell. The host cell then uses its processes to make new viral genetic material and new viral proteins by replicating the virus's genetic material and by synthesizing virus's proteins. And this means that new viruses are made, and they're made through the assembly of the new virus, which involves the genetic material of the virus and the virus's protein joining together and assembling. The virus then lyses the cell, and this means that they can be released to go and infect other cells. And the cell lyses, it damages the cell, and because those cells don't function anymore, it causes a disease. So describe the life cycle of a lytic virus. You can remember this using the acronym SALR. So A is for attachment of virus to specific host cell. Then I for injection of viral genetic material into host cell. Then you've got R and S. So you've got replication and synthesis of new viral components. So in other words, the host cell makes the viral genetic material and it also synthesizes the viral proteins using the host cell's processes. Then you've got A for assembly, and that's assembly of the new viral components. So the viral genetic material and the viral proteins assemble together to make new viruses. And then there is lysis of the infected host cell. And then to finish off, the fully assembled virus is released so that they can go and infect other cells. So in that question might be explaining why viruses can cause disease. That's because they could damage cells, because they take over the cell's processes, and when they break out the cell through lysis, that means that cell doesn't function normally anymore, and that means that the person's got a disease. And a good example of this is HIV. So when we look at HIV, using the acronym, we could say that the HIV virus attaches to white blood cell, injects its genetic material into the white blood cell. The white blood cell then uses its process to replicate the virus's genetic material and to synthesize the virus's protein. And then this viral genetic material and viral protein then joins together and assembles into new viruses. And then the HIV causes the white blood cell to lyse, and then it's released to go and infect other white blood cells. And because there is damage to white blood cells, the immune system is damaged, and that could lead to AIDS, which, as we mentioned earlier, could lead to other diseases as well, such as TB. So this is known as the lytic pathway because it causes the cell to lyse when the virus is released from the host cell. Some viruses can hide within a cell to evade the immune system. So instead of being involved in the lytic cycle, they move into a different cycle called the lysogenic cycle. Here, when the genetic material is injected into the host cell, that genetic material inserts itself into the host cell's genetic material and therefore becomes part of the host cell's genome. We've seen this application before in genetic modification, where we can use viruses as a vector because we could insert our genes that we want for our characteristics in an organism into a virus, and the virus will then infect a host cell which will now contain the gene that will produce the desired characteristic that we want. And then the host cell could divide, and because the host cell divides, because it contains the virus's genetic material in its genome, that means that both cells that are produced through cell division will both contain the virus's genetic material. So this is a way of replicating the virus, not by producing more virus, but by increasing the amount of virus's genetic material, because now there are two cells that have the virus's genetic material. As we learned earlier, that could happen in animal and plants, and can also happen in bacteria, because all three of those cells produce cells that are genetically identical. So if they've got viral genetic material in their genome of the host cell, that means that the daughter cells will also contain the viral genetic material. And this happens obviously from replication of the genetic material. This is known as the lysogenic cycle. And what happens is, after a while, the viral genetic material separates from the host's genome, and when it does that, it moves back into the lytic cycle, where it uses the host cell's processes to replicate genetic material and to synthesize viral proteins. So by moving back into the lytic cycle, you could then assembles new viruses, and those new viruses then cause the cell to lyse and are released to infect other cells. So you can see here a virus has genetic material, which could be DNA and RNA, as is closed by a protein coat, which is the protein capsid. We know that viruses are not living because they don't have processes to replicate, so they use a host instead. So here they are attaching to a host cell, injecting their genetic material, and then the host cell uses its processes to make more viral genetic material and to synthesize viral proteins. They get assembled. After they're assembled, the host cell lyses and the virus is free and it's released so that it can then go and infect other host cells. And that's the lytic pathway. But sometimes when the virus injects its genetic material, that genetic material of the virus inserts itself into the host's genetic material and becomes part of the host's genome. And so when the host cell divides, that means that both cells will contain the virus's genetic material, and this is known as the lysogenic pathway. So in this cycle, new viruses aren't made. Instead, there's replication of the genetic material of the virus, because the host cell is also dividing through replication. Eventually, that virus's genetic material will separate from the host's genetic material and move back into the lytic pathway. So compare and contrast the lytic and lysogenic pathways in a virus life cycle. So you can see in the lytic cycle, viruses enter a cell and cause the cell to make lots of new viruses, which then leave the cell. Whereas in a lysogenic cycle, the virus's genetic material is inserted into the cell's genetic material, so that is replicated every time the cell divides. And that means that each new cell contains the virus's genetic material. Then if you're talking about differences, in the lysogenic pathway, the virus's genetic material is inserted into the cell's genetic material and copied at the same time. But in the lytic pathway, many copies of the virus's genetic materials are made separately from the cell's genetic material using the host's cell processes. In the lysogenic pathway, no new viruses are made, but in the lytic pathway, many new viruses are made. And this is summarized here as well. Now, the effect of viruses on bacteria can be studied using bacterial lawn plates. And these are plates made with nutrient agar. And that nutrient agar has a thin layer of bacteria that grows on the nutrient agar. And that's why it's known as a bacterial lawn plate, because you've got a lawn of bacteria growing on the top of the nutrient agar. You can add a solution of virus to the plate, and after a day when bacteria grows, you see some clear circles. And in these clear circles, the virus has killed the bacteria. So that means there is no bacterial growth in that area. You could calculate the size of those clear circle or the cross-sectional area by using the equation pi r squared, because we're trying to calculate the area of the circle. So r is the radius, you square it and you multiply it by pi. And the larger the clear area, the more effective the virus is at replicating and killing the bacteria. So a larger clear circle means that the virus replicates faster and is more efficient at killing the bacteria. So this is one way of comparing different viruses and looking at their effect on bacteria. So you have your nutrient agar, which is the growth medium that allows bacteria to grow on it. You add viruses and then you give it a day roughly to let the bacteria grow using the nutrient agar, but the bacteria doesn't grow in certain areas next to the virus, and that's because the virus is killing the bacteria. This is known as the zone of inhibition because there is inhibited growth of the bacteria. And then to calculate those clear circles or the zone of inhibition, you use the equation pi r squared. So in this question here, a bacterial plate was infected with two different viruses. After a few days, the radius of the clear space in one of them was 1.2 millimeters and the other one was 1.4 millimeters. So you have to calculate the cross-sectional area, the zone of inhibition. So you can see it's just pi multiplied by 1.2 squared, which is 4.52 millimeters squared, because the units were in millimeters. Whereas in the second one, we've got 6.16 millimeters squared. So you can see in area B, the viruses replicated much faster and killed more bacteria. So it was more effective at doing that because it's got a larger clear area or a larger cross-sectional area or a larger zone of inhibition. Now, humans can defend themselves against attack from pests and pathogens using physical barriers such as the skin, mucus and cilia. We mentioned earlier that in plants there is a disease called cholera ash dieback caused by fungus which causes bark lesions. So how do plants defend themselves against attack from pests and pathogens using physical barriers? Well, plants have an outer layer of leaves that is covered in waxy cuticle, and that waxy cuticle is thick, which means that it's hard for pests and pathogens to penetrate. They also have another physical barrier once they get through if they get through the waxy cuticle, which is the cell wall of the cell. And this is made of cellulose. And that cellulose cell wall is tough, which means that it's hard to break down. So unless you have a cellulase enzyme, it's very tough to break through the cellulose cell wall. So both of these are examples of physical barriers that plants use to defend themselves against attack from pests and pathogens. Unfortunately, physical barriers are not good protection against herbivores. This includes pests such as caterpillars and aphids. So that means plants need other methods of defending themselves as well. So some plants can produce poisons or insect repellents. And these aren't examples of physical barriers, they're examples of chemical defenses. And because plants can produce poison and insect repellents to defend themselves, that means we could utilize that by making medicine out of those chemicals. In your exam, you need to know two types of medicines we can make, which are used to treat human diseases or to relieve symptoms. In your exam, you don't need to know examples of them, you just need to know examples of uses. So we could treat human diseases such as malaria, or we could relieve symptoms such as pain and fever. Now, when making medicines from chemical substances in plants, we have to do tests in labs. And to do those tests, that means that there should be no contamination of microorganisms from the air or from the equipment, and that involves using aseptic technique. So from previous unit, we learnt that if you want to test out chemicals on cells, you'd want to use tissue culture, but we don't want other microorganisms infecting our cells in the plates. So that means we've got to use aseptic technique. So to kill those microorganisms, we've got to make sure that there is sterilization, and that sterilization involves sterilizing both the petri dish and the culture medium, which is the growth medium used to grow the cells. The petri dish can be sterilized by autoclaving, and this involves using heating to a very high temperature. The culture media also has to be sterilized, so culture media such as neutron agar can be sterilized by also heating to a high temperature, and that could also be done using an autoclave. Now, when transferring your bacteria that you're going to test on, or any other type of cell, you need to also sterilize the inoculating loops, because you're using the inoculating loops to transfer the microorganism into your growth medium, the neutron agar. And because you're using the inoculating loop also has to be sterilized, because you don't want other microorganisms to be on that inoculating loop, because they can contaminate and have an effect on the growth of your microorganisms in your plate. You only want your microorganisms to be transferred to the plate. You don't want other microorganisms there, so you've got to make sure you're sterilizing the inoculating loop before you transfer your microorganism that you want to test on. And the way to sterilize it is by flaming the inoculating loop. And when you flame it, the hot temperature will kill any microorganism that's on the inoculating loop. And then once you've transferred your microorganism onto the petri dish that contains the growth medium, you want to make sure that you seal the petri dish so that no microorganisms enter through the air. And you also want to make sure that your vials containing the bacteria are kept covered as well, because that means you won't be just transferring your microorganism, there might be other microorganisms that get into that vial that also gets transferred. So all of these aseptic technique means that only your desired bacteria is transferred to the growth medium, and that means if other microorganisms are not transferred, because you don't want them to affect the spread of unwanted microorganisms. So if you look at the spec, you need to explain the aseptic techniques used in culturing microorganism laboratory. Culturing just means growing those microorganisms, and that means you need to know how to understand the idea of autoclaving to prepare sterile growth medium in petri dishes, and you also need to know the idea of how you sterilize inoculating loops to transfer microorganisms, and the need to keep petri dishes and culture vials covered. So in this exam question here, give 3 ways in which cultures of microorganisms can easily be contaminated with other microorganisms during preparation. They could come through the air, or they could be transferred by you touching the material or any sort of equipment, and they could also be transferred through being in the culture media or in the equipment that you're using if you haven't sterilized them. Describe fully how you'd prepare a culture of microorganisms that you could investigate the action of a plant extract on the microorganism. So we said earlier that we could use chemicals from plants, so if you want to test them out on cells to check that what the effects are on those cells, that means you get your petri dish and growth medium and you have to sterilize them. As we mentioned, we can use autoclave, and then the inoculating loop has to be sterilized by passing it through the flame. Obviously you cool it down before you transfer your microorganism, or in this to grow the dish with bacteria in it. And the loop is then dipped into bacterial culture and used to inoculate the growth medium in the petri dish. And you want to keep the dish as covered as possible, because this reduces contamination from the air. And then after the plant extract is added, the lid of the dish is secured with tape, and that prevents contamination from the air, as mentioned. How can plant disease be detected and identified? So you can see here we've got disease of the plant, but we're going to identify what type of disease it is. And we can identify it first by using observation of the visible symptoms. So you can see here that this plant has a different color leaves, or that some part of the plant is dead. So that gives us a warning sign that there is a disease in the plant, and now we've got to find ways of identifying it. So in these examples here, you might compare it to previous examples of known symptoms. So if you know that a disease is caused by shows the symptoms of a certain color leaves, then you know, or you could identify what disease it is. But there's a problem, because different diseases can produce the same symptoms. So here's an example. We've got 3 different causes of disease, and they all cause the leaves to become yellow. So how do you know what the disease is? So what you could do is you look at the distribution of the damaged plants in the crop, because they change over time. So in other words, a lack of water or magnesium in the soil should affect all the plants in the area at the same time. So looking at the distribution, if you see that all the plants are affected, you know that there's a good chance that it's got to do with something in the soil, such as however, the leaf mold infection is more likely to spread from the plant to another plant that following a wind direction. So you could spot a pattern of the direction of the wind causing the spread of that disease. So this sort of analysis is known as distribution analysis, because you're doing a distribution analysis of the affected plant. You're looking at where damaged plants occur to spot a pattern. For instance, if a disease is spread by wind, it would be more widespread, and this because things like spores can be spread through the wind, so you'd see in a certain direction the spread of the disease. If a disease is spread by soil pathogens, it'd affect a smaller area, whereas if all plants are affected, that means it must be due to an environmental factor such as the soil pH or flooding or drought or deficiency of soil nutrient, because all the plants will be affected. Now if you want to rule out those environmental factors, your third method would be to eliminate possible environmental causes, and that involves changing the environmental conditions. So an example is adding certain nutrients to the soil and checking if the plant symptoms change, because then if they do change, you know that they must have been deficient in whatever nutrient that you're adding to the soil. You could also do something called diagnostic testing in the lab, and that helps you to identify pathogens. This involves taking a sample of the plant that's been affected and analyzing it in the lab, and that means that you might be able to detect the antigens on the pathogen that's causing the disease, or you might be able to analyze the DNA to help identify what the disease is, or what the pathogen that is causing the disease. So you can see in this example, if there's a lack of water, we could check the soil moisture. If it's due to a deficiency in magnesium, we could add magnesium and to see if there's any changes to the plant symptoms. And if it's due to fungal infection, we can grow that fungus or the mold, or we could identify what type of DNA it has and then compare it to other fungus to identify what the cause is. So if you can't identify diseases based on their physical looks, because all 3 examples here produce yellow leaves, there are other methods you use afterwards. So exam question: a farmer finds black spots on the leaves of this crop. Describe how the cause of the black spots could be identified. First, you could check against photos or other information to find out the cause of black spots. You could look at problems in the soil, so take a soil sample for analysis. Check how widespread the plants with black spots are, so that involves distribution analysis, because you're trying to look for a pattern in the infection. And then you could do diagnostic testing, where you send the information or a sample of the plant with black spots to a lab, and the lab can then carry out tests to compare the results to decide what the cause of the spots are. Describe 3 ways in plant disease may be identified. So as we mentioned, is visible symptoms, to look at visibly what changes have occurred in the plant. We can use distribution analysis to see how the problem is spread in the field, or we can do diagnostic testing in the lab, so we identify the genetic material. We could grow the pathogen, which means culturing it, or we could use a microscope to look for the signs that could give us an idea based on previous results. Now the human body could provide protection against pathogens using lots of different methods. So it produces lysosome enzymes in tears, and that kills bacteria by digesting the cell wall. So in the exam, you don't want to just mention that the lysosome enzyme and kills bacteria. You need to describe how. So that means that the lysosome enzyme digests, which means breaks down the cell walls of bacteria, and that's how lysosome enzymes produced in tears protects us from pathogens. We also produce hydrochloric acid in the stomach, and that kills pathogens in food and drink. We have a skin barrier, which is a protective barrier that's too thick for most pathogens to get through. So this is very similar to the waxy cuticle of a plant leaf. And then we also produce sticky mucus in our breathing passage and the lungs, and that traps pathogens. And then that mucus is removed by cilia on the cell lining of the lung and moves like a wave motion, and that moves the mucus that has the trapped pathogens upwards towards the throat, and then we swallow it into our stomach that contains the hydrochloric acid. And we've seen this before from SB1. So you can see in this example here, in the lining of the airway, we've got mucus and you've got ciliate epithelial cells. And then this traps dust and pathogens in the mucus, and the cilia waves and rushes the mucus up towards the throat, where it can be swallowed into the stomach that contains hydrochloric acid. Now we need to know what type of protection they are. So both lysosome and hydrochloric enzyme are examples of chemical defenses, whereas the skin, mucus and cilia are examples of physical barriers. So this is just like how plants are protected. They've got chemical defenses and physical barriers, so do humans. Explain how sexually transmitted infections, STIs, are spread in chlamydia and HIV. Chlamydia involves a bacterial pathogen, whereas HIV, we mentioned earlier, is due to a virus. The STIs are spread by contact with body fluids such as semen and vaginal fluids from an infected person. So how are they transmitted? An infected mother can pass chlamydia to a baby during birth, so that means that it's due to contact through the vaginal fluid during birth. Whereas HIV transmission occurs through unprotected sex with an infected partner. It could also be due to transmission from an infected mother to the fetus, or infection from blood products that are used to give to a person that requires blood. So how can we reduce or prevent the spread? So in terms of unprotected sex, just use condoms during sexual intercourse. To reduce or prevent transmission from an infected mother to fetus, we could screen people, especially pregnant women, and then we could give them treatment. So if it's chlamydia, they could have antibiotics before they give birth, so hopefully it won't be transmitted when they're giving birth to their baby. In terms of infection from blood products, we could screen the blood transfusion so that they're not giving to people that require blood. That blood will not contain pathogens. Name one protist pathogen that's able to get through the skin barrier and explain how it does this. So this is Plasmodium, which is a protist. It causes malaria. It gets through the skin barrier when it is injected directly into the blood by the mosquito animal vector. Explain why lysosome is said to be a chemical defense of the body, because it reacts with the bacterial cell wall and causes breakdown, which kills the pathogen. Is mucus a physical barrier or chemical defense? It's a physical barrier because the pathogens have difficulty getting past it to the cells. And then how does the cilia help cells lining tubes in the lungs carry out its function? What it does is it has extensions called cilia. Those ciliate epithelial cells have cilia on the surface that moves the dust and pathogens out of the lungs, and that reduces the risk of infection because it moves it up to the throat, and then the pathogens that are trapped in the mucus are swallowed into the stomach, where they're killed by the hydrochloric acid. Explain why the stomach is a good defense against pathogens. That's because many pathogens get into the stomach through food and drink. This is oral transmission, but they're destroyed by the hydrochloric acid, the low pH in the stomach. So this is a chemical defense against infection. And then how does screening for an STI help to reduce spread of infection? Screening identifies people who are infected with STIs They can be treated for the disease, and they will also know they should not have unprotected sex until they're clear of the infection, and that reduces the spread of the STI to other people. And then sexual partners can also be screened to check if they've got the infection as well. Now, unfortunately, sometimes pathogens manage to get through both the physical barriers and the chemical defense of the body, and therefore now it's the job of the immune system to deal with those pathogens, to attack them and try to prevent them from causing harm. All cells and viruses have molecules on their outer surfaces called antigens. So you can see here on the outer surfaces of the bacteria there are antigens, and on the virus as well there are antigens, and you'll notice that these antigens are different. So different pathogens have got different specific antigens on their surface, and it's also in all types of cells. So it doesn't have to be a pathogen. You can find them in red blood cells. You can see that the type of antigens on the surface are different, and that means that we've got different blood groups, as we discussed in the previous units. So how does the immune system deal with pathogens? As we mentioned, the pathogens have antigens on the surface that are unique to them. So that means there are lots of different types of pathogens, and they all have different antigens on their surface. So that could help us recognize what type of pathogen they are. And then the immune system involves the white blood cells called lymphocytes, and those lymphocytes have got molecules on their surface called antibodies. There are lots of different types of lymphocytes, and all of these lymphocytes have got different antibodies on their surface. Because they've got different antibodies on the surface, what happens is that those lymphocytes with antibodies on the surface have a matching shape to the antigen and the pathogen. So that means a specific lymphocyte will have a specific antibody on its surface that matches the unique specific antigen on the surface of the pathogen. And when it binds, that lymphocyte will be involved in stopping the pathogen from attacking the body. So once it binds and the correct or the specific antibody matches or binds to the antigen of the pathogen, this activates the lymphocyte to divide rapidly, and that produces lots of identical lymphocytes. That produces the antibodies that are going to be used to damage the pathogen. And then some of those lymphocytes secrete the antibodies, which then stick on to the antigens and destroy the pathogen. And then those that remain in the body are known as memory lymphocytes, and then they're used in the future in case the body is infected by the same type of pathogen. And this means that someone is ready to respond immediately if the same antigen or pathogen enters the body again. So when all the pathogens have been killed, some of the lymphocytes with antibodies that match the pathogen's antigens remain in the blood. They're called memory lymphocytes. If the same kind of pathogen tries to infect again, the memory lymphocytes cause a much faster secondary response that will stop you becoming ill. That means that you are immune to that pathogen, because the chance of you now getting infected or having a disease is less, because you have got these memory lymphocytes which are ready to deal with having the same pathogen infecting you. And this secondary response, as we mentioned earlier, is faster and produces more antibodies than the primary response, and this kills or stops pathogens before there is enough of them to make you feel ill. So actually it's very likely that we're continuously getting infected with pathogens, but we don't feel ill because we've got a secondary response through memory lymphocytes. That means that we don't actually feel ill because it responds faster and produces more antibodies quicker than the primary response. So to summarize all that again, different lymphocytes have got different antibodies, and when a pathogen enters the body to infect, it will have its specific antigen on the surface of the pathogen. The different lymphocytes will then try to bind onto the antigen of the pathogen. Remember, there are different lymphocytes with different antibodies, so you want the lymphocyte with antibodies that matches the antigen on the pathogen. And when it does that, it then gets activated When that lymphocyte is activated, it replicates rapidly through cell division, and then some of these lymphocytes produce antibodies that are released. An antibody can only bind to the antigen that caused it to be produced, so that means they're very highly specific, and each different type of antigen causes a different type of antibody to be produced. So the production of these antibodies that are highly specific that match the antigens of the pathogen causes damage to the pathogen, and then some of these cells stay behind. They're known as memory lymphocytes. Once the pathogen has been destroyed, a few memory lymphocytes remain. They recognize the pathogen in the future if there's reinfection, and the immune response is much quicker and more effective. And that's because there is a faster second response which produces more antibodies, and that kills or stops the pathogens before enough of them make you feel ill. Now we could take advantage of this through immunization. So immunity to pathogen immunization can be triggered artificially by using a vaccine, and that's known as vaccination. And the vaccine contains weakened or inactive pathogens, or it could also include just the antigens only. So instead of having a pathogen infect us, we can inject weakened or inactive pathogens or the antigens of the pathogen into our body, and these weakened or inactive pathogens or the antigens won't necessarily give us the disease, but it means that our immune will respond to it. So that means that the person's lymphocyte will produce antibodies against the pathogen, and that produces the memory lymphocytes, and then those memory lymphocytes are used in the second response very rapidly, and that means that the person is very unlikely to become ill without even having the infection in the first place. So there's no primary response, there is just a response straight away due to vaccination. Now there are lots of advantages and disadvantages of these. Clearly the advantage of immunity is that it's produced without being ill, it lasts a long time, so you're getting protection through immunity. But the disadvantage is some people get an allergic reaction, so they can't take the vaccination, and then very rarely a person has a major harmful reaction to it. One big advantage is if most people are immune, then few people that can't get vaccinated are less likely to catch the disease. This is known as herd immunity because the vast majority of people have had the vaccination, so that means that they're protected through immunization. So those that can't have the vaccination are protected because there are less people that are likely to have and spread the disease. So to summarize that in an exam question, a pathogen gets past the body's natural defense through a wound in the skin. So they've passed obviously both the physical barrier and the chemical barrier as well. And then in the blood, the pathogen comes into contact with white blood cells called lymphocytes. Then specific lymphocytes have specific antibodies on their surface, and one of them will match the antigens on the surface of the pathogen, and this lymphocyte will get activated, and it then divides rapidly to produce lots of identical copies of that specific lymphocyte. And then some of those lymphocytes that are produced through cell division will release a large amount of antibody into the blood, which will destroy the pathogen. And then after the pathogens are destroyed, some of the lymphocytes remain in the blood as memory lymphocytes. This means that if you're affected again with the same pathogen, the memory lymphocytes will produce a much more rapid and larger immune response, and this is known as a second response, where it's a quicker response and it produces antibodies much faster as well. So exam question, explain how immunization of the majority of children protects all the children in the community from these disease. So inactive forms or antigens or pathogens are given to the child, then the antigen of the pathogen cause an immune response and triggers response by or activate lymphocytes in the blood that match the antigen. The activated lymphocytes divide rapidly to produce more lymphocytes or release antibodies to the antigen. Some lymphocytes become memory lymphocytes that stay in the blood after immune response ends. Then exposure to the live pathogen triggers a second response. So this means if you get infected by a live pathogen, you now have a second response which produces large amounts of antibodies more quickly than a first exposure to the pathogen which happened through vaccination. The second response prevents illness, so the person is now immune to the disease. And then if immunization of the majority of group makes it unlikely that those that can't be immunized are very likely to get the infection, and this is known as herd immunity. And then there are additional exam questions here that you can read. So this is what the spec says that you need to know. So moving on to antibiotics. Explain why antibiotics can only be used to treat bacterial infection. So a common exam question would be, why can't it be used to treat viral infections, or why doesn't it affect human cells? So that's because antibiotics inhibit the cell process of the bacteria, and that stops the bacteria from growing or reproducing. It doesn't affect humans because the human cell processes is different to the bacterial cell. So that means the human cells processes are not inhibited by the antibiotics. It's only the bacterial cell processes that are inhibited. That stops the bacteria from growing or reproducing. Now Fleming discovered antibiotics known as penicillin to describe the process of developing new medicines, including antibiotics. So there are 4 stages. The first stage is discovering the new medicine. And that could be done through screening organisms to see if they produce antibiotics that kill bacteria, and then they are developed through a series of stages afterwards, the other 3 stages. Remember, in discovery we know about plants producing chemical defenses, so we could extract plant chemicals to use as medicine, and that links to the previous topic that we discussed. So plant chemicals can be used as medicines to treat human disease or relieve symptoms, and that involves through discovery of the plant chemical extract. The next stage is the preclinical testing, which is done in the lab, and that involves testing first on cultures of cells. So you can test it on human cells or plant cells or bacterial cells. You could also test it on their tissues, and the next stage of preclinical testing would be to test it on animals. So the antibiotics are tested in the lab to make sure the medicine gets into cells without harming them and damages the pathogens inside the cell. In the exam you need to explain this. You can't just mention that you do a preclinical test on cells, tissues or animals. You need to mention the fact that you're doing this because you want to check that the medicine gets into the cells without harming the cells, and it also you want to check if it damages the pathogens inside the cell. The third stage is the clinical trial, which is split into 2. So the first clinical trial is done on healthy volunteers. You have a very small dose of the drug, and this is to check that the drug is not toxic or harmful to those healthy volunteers. Then the next clinical trial involves stage 2, and that's because different doses of drugs are given. So you're testing to find out the optimum dose of the drug. You want to test which dose works the best. In this case, you're checking for different side effects on different people. So this is done on a large group of disease patients, and you want to see what response those different groups, whether it's age, gender or ethnic group, have based on the drug. So clinical trial stage 1 and stage 2 are different because in stage 1 you are trialing on a small number of healthy volunteers to check that the drug is not toxic, check if it's safe. You could also check what sort of side effects there are, just to make sure that there are small side effects. Whereas in clinical trial stage 2, you're testing it on a large group of disease patients to find out what the different side effects are on different people and to see what response of different groups there are. List the stage of developing a new medicine, explain why each stage is needed. So like I said in exam, you can't just describe or list the stages, you need to explain why each stage is needed. So we know that the stages are discovery, preclinical stage where you're testing on cells, tissues, and then animals, and then you've got 2 clinical trial stages, a small scale on healthy people and a large scale on diseased people. And then why is each stage needed? You test on cells, tissues or organs to make sure that the medicine affects the disease but doesn't harm the cells. So you want to check also that it could go into the cells. When you test on animals, you make sure the medicine works on a complete body without harming the whole organism. And then on a clinical trial, you test on a few healthy people to check that the medicine is safe, and you're also looking for what sort of side effects there are. And then the last clinical trial involves testing on a large number of people, but these people are diseased people, and this is to make sure that the medicine helps them get better. It also identifies the right dose, and it looks at a wide range of side effects in different people. Now, when you're doing testing in your exam, you need to understand how to investigate the effect of antiseptics, antibiotics and plant extract on microbial cultures. And this means that you need to understand that the independent variable would be the type or concentration of those three things. The dependent variable would be the zone of inhibition, in other words, the size of that clear circle, which is calculated through pi r squared. Control variables would be the volume of the antiseptic, antibiotic or plant extract. Another control variable, which is the one that you should most likely write down, is the incubation temperature, because when you're growing the bacteria on the cell plate, you want to use the optimal conditions, such as the same temperature, also the type of agar or the amount of bacteria that's spread in the agar plate should be the same. And then you can do a control experiment as well, because you want to use a paper disc that doesn't have these antiseptic, antibiotic or plant extract, and this should result in no zone of inhibition, and that proves that the clear area around the other disc is due to the antibiotic or the antiseptic or the plant extract and not something else in the disc. Risk assessment that you need to do is make sure you use gloves and safety goggles, and that prevents infection and contamination. So remember, we mentioned that aseptic techniques have to be used. We've already discussed how to calculate the clear circles using pi r squared and why you have clear circles. If you want to check or find out more information, you could look at this YouTube site. Now, to lawn the bacterial plates, you have to prepare them, as we discussed. We're using nutrient agar plates, where you have a thin layer of bacteria that grows after a day by growing it on optimal conditions. You add your paper disc, and then you incubate the bacterial lawn plate at the optimum temperature, and you measure the zone of inhibition, and you check to see how the bacteria has been killed, or how effectively it has been killed by the antiseptic, the antibiotic or the plant extract. You could also even mention viruses if the questions are about the virus's effect on bacteria. And here's a summary of the investigation. Okay, and the last topic is about monoclonal antibodies. Monoclonal antibodies are identical. So mono just means one, only or single. That means it's the same. And clonal is identical. So in other words, monoclonal antibodies is when you're trying to produce a large amount of the same antibodies. Now, we know antibodies are produced by white blood cells. So if you're trying to produce a large amount of antibodies, you need white blood cells. And you wouldn't take these from humans, so you'd want to use an organism such as a mouse. The advantage is that if you get white blood cells, or the lymphocytes, they could produce the antibodies. The problem is, once lymphocytes produce antibodies, they can't divide. So that means you can't produce large quantities. So you want a cell that could divide continuously. One cell that divides continuously or uncontrollably are cancer cells. So the advantage of cancer cells is that it continuously divides. But the problem with cancer cells is they don't produce antibodies. So you've got lymphocytes that could produce antibodies, but they don't divide, which means that you can't produce large quantities. And then you've got cancer cells that could divide continuously, so that means you have lots of cancer cells, but they don't produce antibodies. So to produce large quantities, you want to fuse the lymphocyte with the cancer cell. And this produces hybridoma cells through the fusion of the lymphocyte and the cancer cell. The hybridoma cell has the properties of both the lymphocyte and the cancer cell. In other words, it could divide continuously like the cancer cell, and at the same time it could produce antibodies as well, just like the lymphocyte. So monoclonal antibodies cannot be made in large amounts using normal lymphocytes. This is because a lymphocyte can divide over and over again to make copies of itself clone, but once it produces antibodies, it can't divide anymore. So we can't use lymphocytes to produce large quantities. So to get around this problem, we produce hydroma cells by fusing a lymphocyte that produces the antibodies that we want with a cancer cell. And the cancer cells are useful because they can divide over and over again. So that means the hybridoma cells have the characteristics of both the lymphocyte, which produces the antibodies that we want, and the cancer cells, which means that they can divide over and over again. So here are the stages which you need to write down for your exam. First, you need to have a particular antigen and you inject that into the mouse. Because there is an antigen, the mouse's immune system responds. When it responds, it produces lymphocyte cells that are activated and they divide continuously against the antigen that's been injected. So now you've got lots of lymphocytes from the mouse, and you could then fuse it with cancer cells that you grow on culture media. So this is like tissue culture. And when you fuse them together, you produce hybridoma cells. And that hybridoma cells can both divide, because it's got the properties of the cancer cell, and it can also make antibodies of based on what you use or injected into the mouse. And this is now produce, or these hybridoma cells will produce monoclonal antibodies.
Consequently, they are highly specific and have applications in diagnostic microbiology and cancer therapy. The first step is to inoculate an animal with the antigen carrying the specific epitope. B cells are then isolated from the spleen and then mixed with a type of cancer cell called a myeloma cell that grows continuously. The two types of cells fuse together to form cells called hybridomas. The mixture of cells is cultured under conditions which permit growth of the hybridoma cells, but not the B cells or the myeloma cells. The hybridoma cells are then separated into individual chambers and tested for ability to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured, and monoclonal antibodies are isolated and purified.
Okay, if you look at the spec, you need to describe the production of monoclonal antibodies, and as we mentioned, using lymphocytes will produce the desired antibodies, the production of hybridoma cells, and then those hybridoma cells producing antibodies as they divide because they got the properties of the cancer cells. And then a much harder question would be explaining the use of monoclonal antibodies, including in pregnancy testing. So in pregnancy testing, you could identify if someone's pregnant because they produce a pregnancy hormone in their urine, and the monoclonal antibody matches the hormone and causes a reaction in the indicator that indicates that that person is pregnant. So how could you produce monoclonal antibodies needed for pregnancy stick? Remember, you're trying to make monoclonal antibodies, so you want to make a large quantity of the same type of antibodies that will tell you if someone is pregnant. So what you do is you inject a mouse with the human pregnancy hormone, and the mouse will then react with using its immune system and produce lymphocytes specifically for the human pregnancy hormone. And then you fuse that lymphocyte with the cancer cell to produce hybridoma cell. So the lymphocytes that make antibodies matching the human pregnancy hormone taken from mouse and fused with the cancer cell, and now that means you produce hybridoma cells that could continuously divide. So that means you could make large quantities of those cells, and they can make lots of antibodies that match the human pregnancy hormone, and you can put that on the strip. So here you can see that the second line indicates a positive test. You put your monoclonal antibodies which you just made using the method just described, and then when a female pregnant uses her urine on the pregnancy strip, the hormone that they produce will bind onto the monoclonal antibodies that was produced from the mouse, and that will indicate that that person is pregnant. Monoclonal antibodies can be made to match and stick to any type of protein such as hormones and enzymes. They can be made to match the antigens and pathogens as well, and they can also be made to stick to specific cells in the body such as cancer cells or platelets, which form when blood clots, which we mentioned earlier could lead to things like heart attack and stroke. So what's the method for doing that? Well, you just get the cancer cell antigens or the platelet antigens and inject that into the mouse. The mouse will respond by producing specific lymphocytes which are activated because of these antigens, and you fuse them with the cancer cell to produce hybridoma cells. And then these monoclonal antibodies can be made to stick to certain types of cell used in medical diagnosis. So you could use a PET scanner to find out the position of the cancer, because you've now produced monoclonal antibodies that bind onto cancer cells. And when you use a PET scanner, which we mentioned in a previous unit, which involves radioactive monoclonal antibodies in this case, you could attach cancer drugs to those monoclonal antibodies, and they will directly deliver the drug to the cancer cells to have an effect on these cancer cells and kill them. And this reduces the risk of damaging healthy cells, because you don't have to use radiotherapy, which kills actively dividing healthy cells, or use chemotherapy, because those drugs target all the healthy cells in the body instead of directly targeting the cancer cells, which can be done by attaching cancer cells to monoclonal antibodies. So you can see here, explain the use of monoclonal antibodies. You need to understand how to do it in pregnancy testing, in diagnosing a patient, including the location of the position of blood clots and cancer cells. And then you need to also know the advantage of using monoclonal antibodies to target specific cells compared to drug and radiotherapy. So here's some exam questions with answers that you should read. And that is the whole of SB5.